Great. Well, I guess I'll just start by thanking everybody for and the organizers of the meeting for inviting me to give this presentation. Yeah, I attended all the talks yesterday, and it was really interesting. And unfortunately, I didn't get the chance to come to Vienna, but hopefully that'll happen one day soon. Um, so I figured for this talk, I would just sort of start off with just covering some general work that we've been doing on various Bayesian methods for phylogenetic analysis of both fossil and extant taxa. So for much of the history of the field of systematics, we've really relied on estimating phylogenetic trees whereby the branch lengths are proportional to the number of substitutions per character. And this is really through the motivation to build phylogenies where we can understand the relationships of fossil taxa or various taxa in relationship to the amount of genetic or morphological divergence among those species. However, as evolutionary biologists, we're much more interested in really getting a sense of the evolutionary timescale of speciation events on an absolute or geological scale. And that's because we're really interested in determining the sequence of these events, how they happened in time, and sort of the rate and tempo and mode of those of evolutionary processes. And so because we're much more interested in these types of representations of phylogenies, because they give us much more information, there's been a lot of work in recent years really working toward integrating information that gives us that we observe, and we can calibrate and scale these trees to an absolute time scale. So the fossil record really is our primary source of information for placing phylogenies in this absolute time scale, where the branch lengths are proportional to years or millions of years. And essentially what fossils are, they are snapshots of a phylogeny that's been preserved over the history of Earth. So a single fossil specimen can offer loads of information about the geology, ecology, and taxonomy of a particular clade. And so with this information, we can use phylogenetic methods to understand how fossils are related to other taxa and recovered from the fossil record, related to other taxa recovered from the fossil record, as well as to taxa that are present today. And so this cartoon just shows an unrooted phylogeny of a tree with a single fossil species and 5 other taxa, including an outgroup. And so for a long time in phylogenetics and systematics, we primarily use methods like parsimony to integrate fossil species within a tree of that comprised extant or other fossil taxa. But ultimately, these approaches don't really give us a way to understand the timing or the processes producing these phylogenetic patterns. But with those trees, we can integrate the paleontological information in such a way where we can impose a time scale on the tree. And so classically, these methods will take the fossil record and those phylogenetic relationships and rescale the branches such that they are essentially based on the age of the oldest descendant of a given node. So in this case, the species relationships are integrated into this time scale, but ultimately, because we don't have a process informing or allowing us to infer those divergence times, the branch times don't really are unlikely to really represent the true speciation ages of these nodes. But what we can see when we do integrate our parsimony-based phylogenies into these timescale methods is that that what we're trying to say about these trees and these taxa is that they are all part of this singular process that generates a phylogenetic tree with extant taxa and with fossil species. So because of this, more recent methods for dating phylogenies have really used fossils to calibrate nodes in a tree. So if we can actually root a phylogeny of both living and fossil taxa, we can then identify a total group to which that fossil taxon belongs. And so then once we have defined this total group, we can use this fossil to essentially constrain the age of the node representing its ancestor in a tree of extant species. So this gives us a way then to structure and scale our extant phylogeny or a phylogeny with a fossil taxon. And in the case that if we don't have character information to really place the fossil in a precise place on the tree, given that we can use taxonomy to assign it to a total group, then we can use this occurrence time to essentially constrain the age of that tree and then propagate that information to the rest of the tree. So in about 2 decades ago, I guess, several Bayesian methods were then introduced, or statistical methods were introduced to really get at approaches to integrate these fossil occurrence times to better calibrate phylogenies of extant taxa. And so in these methods, divergence time estimation, in these methods, fossil data was introduced that allowed this to constrain the tree and constrain ages using fossil information as minimum ages. So these methods ultimately involved applying parametric distributions to calibrate the age of ancestral nodes in the tree that are ancestor to the fossil's descendants. So here, the calibration density is offset by the minimum age of that's provided from the fossil record. And so this gives us an approach to estimate the absolute ages on our tree. However, these absolute ages are really driven by our choice in these calibration densities and the parameters we choose for them. And ultimately, what these node calibration approaches kind of put us in a situation where we really actually don't have the ability to use a lot of the information that's provided by the fossil record, because only the oldest descendant of a given node is useful as calibration, as a calibration constraint. And so this leads us to seek ways where we can better integrate more of our the information from the fossil record to get at estimating ages of trees with extant taxa. And so it really is because of what we understand about these fossil species, that they are part of this overall diversification process that gave rise to our tree of extant taxa. Then what makes sense is that it's much more appropriate to represent these fossil species as fully integrated into this diversification process. And so when we initially started working on this methods, we found that these node calibration approaches were a bit unsatisfying. And so this really started a long-term collaboration for me with Tanya Stadler to develop better approaches to calibrate trees to absolute timescales. And so when we think about this birth-death process and what we're trying to do with statistical approaches that are assuming this model, we want to say that the fossil taxa are fully part of this process. So when we apply a birth-death process as a tree prior in a statistical model, well, we have a process that gives rise to a phylogeny with extinct lineages and various speciation times. So this is an animation that's showing the generation of a birth-death tree, and we have some rate of speciation that gives rise to new lineages, and the death rate is the parameter that controls the rate at which lineages go extinct. So here we have this tree where all of the living taxa at any given point in time remain stationary, and then all of the ancestral lineages sort of march away into the past. So here we see the lineages terminated as extinction events happen, and speciation events as speciation, and divergences as speciation events happen. So we can now consider if during this process there's some other process that's allowing lineages to be preserved in the fossil record and eventually recovered by paleontologists. And so if we have such a process, we might get lineages that are preserved in our rock record, and these would occur, you know, sort of as snapshots of this process back in time. And so this is what we're essentially assuming for this serial sample birth-death process. So in 2010, Tanya Stadler had a paper where she introduced a serial sampled birth-death process that gives us a phylogeny where we have fossil timestamps on the tree over time. And so this model assumes that this process generates this tree with these historic time samples, and so that ultimately the process gives us this complete tree. And so here you see that we might have some sampling process where we can sample some of the living taxa and also have a fossil recovery process that gives us these snapshots on the tree. But what this also indicates is that there are going to be lineages because of our sampling process that will be lost to time and never observed. And so a lineage that goes extinct, if it doesn't have sampled descendants, then that lineage will be unrecoverable even by any of our methods. And so what we have from this model described by Tanya Stadler is that the sampled tree, this tree that is defined by our set of samples, is a process that's extracted essentially from this complete tree. So then we can calculate the probability of our sampled tree given that it comes from this complete process. So early on, this approach was really used a lot more for serial sampled data sets of infectious diseases, but in our collaboration, we sought to use this for data sets comprising both fossil and extant species. So in the case when we apply this model to a data set that includes fossil taxa, we essentially termed this the fossilized birth-death process. And essentially this name of this model, really, the credit for that does go to Bastien Bousseau, who came up with that because it was obvious, and even though it wasn't obvious to us when we were thinking about what we should name it. So when in our initial application of this model to this data set of fossil and extant taxa, we really sought to use it as an approach to calibrate a tree with extant taxa, but while also using more of the fossils that we had access to. And so essentially what we showed in this paper that we did in collaboration with John Holstebeck is that this approach then gives us a much more statistically coherent way to estimate the divergence times, absolute divergence times of a tree of extant and fossil taxa. But we also have this better way of capturing our statistical uncertainty in these divergent states, because as we incorporate more fossils into our analysis, we get a reduction in our uncertainty. We get more precise estimates of species divergence time. And additionally, this approach does allow us to use much more of the fossil record in these analyses to date phylogeny. So the fossilized birth death is a stochastic process that gives rise to phylogenetic trees of that are scaled proportional to time with fossils sampled over time. And so this is a probabilistic graphical model showing the parameters of this process that gives rise to our phylogenetic tree. So in this model, we have parameters that control various aspects of the process. The process is conditional on the origin time of, so the very start time of the process. And then we have rate parameters that determine the rate at which which lineages diverge, so speciation rate, extinction rate for the rate at which they go extinct, and a rate of fossil recovery that accounts for the rate at which fossils are preserved in the fossil record and then recovered by paleontologists. And additionally, we also have this parameter that accounts for the probability that that only a subset of the living taxa are included in the analysis. And additionally, we have we are able to observe from this process also the fossil occurrence time. So we can take what we understand is that our observations of this process, especially of the process that happens but before the precedence, really comes from these snapshots that are the sampled fossil occurrence times. So as we've begun to implement this type of model into various Bayesian implementations, we have now better approaches for jointly estimating the tree topology and the divergence times when we have data sets that contain both fossil and extant taxa. So these data sets give us, for these data sets, we typically have a morphological matrix that contains discrete morphological characters for both the living and the fossil species. And you can see that we often have quite incomplete morphological data for our fossil taxa. And then we can also integrate this with models accounting for our molecular sequence data. And obviously for our fossil taxa, especially for very ancient ones, we don't have any information that these particular species can contribute to our molecular sequence alignments. But given these data sources, as well as the times associated with our fossil taxa, we can then accommodate uncertainty in the phylogenetic placements of fossils, while also allowing for more information from the fossil record to inform the divergence times in our analysis. So these combined evidence phylogenetic analyses allow us to essentially extend the set of models that we consider. And so here I'm showing a more coarse-grained view of a probabilistic graphical model that's describing sort of the overall process that gives rise to our various data sets. And so this is describing this generating process that's responsible for giving us these sets of observations. And so we have models associated with each of those. So we have a substitution process and rate models associated with our molecular data. Likewise, we have a discrete character morphological model and sets of rate models for giving rise to our morphological characters. And these two, our character data sets, are really linked because we assume that they came from the same overall time tree. And the generating process for that time tree is the one that also gives rise to our fossil occurrence observations. And so here is where we can fit in this, the part of the model that is the fossilized birth death process. So in our combined, with this access to these analysis approaches that allow us to combine all of this information, we can then much more better capture the diversity of certain groups, particularly when they have rich fossil records, and hopefully and intentionally learn more about their overall macroevolution. And so we did this in a project that we collaborate on with Sasha Gavrishkina and Dan Sepka. And we sought to determine if our understanding of evolution in the clade comprising all of the penguin species can be improved when we consider all of the fossil taxa represented in this group. And so here's just a preliminary tree giving a description of the diversity that we see in the penguin fossil record and in the extant crown group. And so what we do see is that much of the diversity in this group really lies on the stem lineage leading up to the most recent common ancestor of all living penguins. And so if we were to only consider the extant taxa in our traditional node calibration approaches for phylogenetic divergence time estimation, we would only ever really consider a small subset of the fossil record and really lose the diversification history that is represented in all of these stem fossils. And so because of that, with these combined evidence approaches using this fossilized birth death method, we can then integrate all of this fossil diversity into our analysis. So in our analysis, we can use this approach to jointly estimate the phylogenetic relationships of all 19 living taxa in the penguin clade, as well as 36 fossil species. And so for this, we had over 200 morphological characters, and we use an alignment of molecular data. So these illustrations just show some representatives of extant species and some representatives of sort of reconstructions of fossil species. So estimation using an integrated set of models then enables us to get a much broader picture of the evolution of penguin diversity over time. And so, this is particularly the case when compared to if we were just to look at this crown group, which most of the previous studies had done. So this tree summarizes our analysis, and the information that we glean from this really allows us to get a much better picture in terms of the placement, the relationships of the fossils, the stem fossils with respect to the crown taxa, as well as the overall timing of various speciation events in this tree. And ultimately, what we do see is that the information that we get from these fossils does inform aspects of the crown group. And this study showed a timeline for the evolution of penguin species over time that was much more consistent with what had been observed in the fossil record. And previous studies that only considered the crown group really didn't match with our understanding of the fossil history, particularly in the crown. So that study was published in 2017, but even as we were doing those analyses, there were more penguin fossils that were essentially waiting to be described. And so this includes this new penguin species called the dawn-crested penguin. And these taxa, what we understand from the fossil record is because they provide such a wealth of fossils, provide such a wealth of information coming from various angles, is that this had the power to really give more insight into the evolutionary history of penguins. So this is a study that was led by Daniel Thomas and Dan Sepka, and they and their paleontological colleagues were working on describing this new dawn-crested penguin. And so what's particularly interesting about this penguin is that it had a lot of key characteristics that linked it taxonomically to the genus Eudiptes. And however, its age and some aspects of its morphology indicated that it was a really early representative of this genus. Additionally, it was one of the earliest fossils that had been discovered from this part of the world that is essentially a seabird hotspot, the Terua Maui area, where about a quarter of the world's living seabirds go and have, essentially it's their breeding ground and nesting ground. And so one of the interesting things about this particular fossil was that it had the ability to sort of unlock the timing of this area for becoming the seabird hotspot. Additionally, in their description of this fossil, Daniel Thomas and Dan Sepka recognized that this dawn-crested penguin, while its bill morphology was really similar in overall shape to other Eudiptes species, the width of this of its bill is much more slender than the living taxa. And so this was indicative that there might have been some sort of overall processes dictating a change in the overall width of the bill mandible for Eudiptes species. And so this led to additional hypotheses about the ecological context in which this particular genus evolved. So for our analyses, what we did was we extended our morphological matrix to include 8 newly described fossil species, and including the dawn-crested penguin. And what what our results show is that this deep bill of the dawn-crested penguin evolved relatively recently, about 2 to 5 million years, or the deep bill of the Eudiptes penguins, evolved relatively recently, about 2 to 5 million years ago. And essentially, going back, just to what's really kind of interesting about this, the evolution of the bill morphology in this genus is that what we see is that the slender bill appears to be a much more ancestral trait that's more similar to the Megadiptes genus, and whereas the living members of this genus are all feeders on particular species of krill, and what's interesting is that this bill morphology is potentially associated with the shape of the tongue. And so this has really led to more hypotheses that can be potentially better understood in terms of the functional morphology of this bill morphology and the ecology of this genus. And additionally, this analysis that provided us with much better dates for understanding the timing of the recruitment of these seabirds to the avifauna of Terua Maui, which occurred in approximately in the late Pliocene. So all of these combined analyses that I've shown for these fossil penguins really demonstrates the utility of these methods for understanding the timing of species diversification. And while the fossil record really is our primary source for understanding the timing of speciation events on this absolute timescale, it's really critical for us to understand that the fossil record is heavily structured and our observations from it come with quite a lot of uncertainty. But fortunately, Bayesian approaches provide us with ways to accommodate this uncertainty, particularly in the fossil occurrence ages. And so previous studies have looked at this when we develop these methods as approaches to accounting for uncertainty when we have a particular fossil taxon with an unknown or highly uncertain age. However, we're interested in better capturing the structure of the fossil record. And what we recognize from the nature of the rock record is that there's often preservation biases and other challenges with fossil dating methods that can cause structured patterns in the uncertainty associated with fossil ages. In particular, it's not uncommon for data sets of fossil taxa to have specimens represented from different fossil formations. So for many groups, only the age estimates of these specimens, or for many groups, the only age estimates for these specimens really comes from relative dates that are calibrated to the age of the entire formation. Thus, you may have a sample of some fossils from deposits that are have really precise age estimates, and others where this age estimate is much less certain. And so such deposits may be really rich and well-preserved fossils, but highly uncertain ages associated with the formation will essentially make it difficult to understand the context of the species preserved in these types of deposits. So this can really then in turn make it difficult to also inform hypotheses really concerning, the evolution, biogeography, or ecology of these lineages observed in these types of formations. So this led to a study that's led by João Barreto-Satani and Dagmar Joá, who are interested, where we're interested in determining the accuracy of these combined evidence approaches for estimating the ages of poorly dated fossil deposits. And in particular, we're really interested in our ability to recover reliable age estimates for the specimens trapped within these types of formations. So to do this, we're working on a simulation study where we simulated trees and fossils under the fossilized birth-death process to determine the accuracy of our age estimates within a hypothetical poorly dated deposit. And so we selected an age uncertainty interval that's associated with this poorly dated deposit. However, not all of the fossils in this time interval are going to be preserved in the deposit. And so to do this, to assign fossils to the deposit, we then used an approach where we simulated association with that deposit based on a model that whereby more closely related fossils would be more likely to be in the same deposit. So then we essentially assigned fossils within our poorly dated deposit time interval to the deposit. And then for any of the fossils associated with the poorly dated deposit, we gave them a time range, an age range that was the entire width of that deposit. And so this is as a way to essentially emulate the nature of the fossil record for many types of fossil data sets. And so for all of the rest of the fossils, we consider these essentially well-dated fossils, and then we assign them an age range that was proportional to their true age. And so for our simulation treatments, we then varied the relative age range of these well-dated fossils, as well as the proportion of the poorly dated fossils in our data set. So in assessing analyses of these simulated data sets, we considered we evaluated the coverage, which is the proportion of our estimates where the 95% credible interval overlaps with the true value. And so what this plot is showing is for all of the well-dated fossils, so the fossils where the age range is just proportional to the true age of that fossil, we're showing the coverage, the proportion of estimates where the true age is covered by that 95% credible interval. And this plot is showing that as we decrease the precision of the age range for the well-dated fossils, we get a predictable decrease in the coverage proportion for those the ages of those well-dated fossils. So this is exactly what we would expect. And so what I'm showing here is this is for the case when we have just 10% of the total fossils fall within this poorly dated formation. And so as we increase the proportion of those poorly dated fossils, we also see a predictable decline in the coverage proportion for the well-dated fossil ages. And so this is just to show that we have this predictable behavior of the method as we increase or decrease the amount of information or the precision of our information, we get an associated increase or decrease in the coverage of our estimator. But we also assess the accuracy of the mean age estimate for these well-dated fossils. And what this plot is showing is the relative error, which is essentially the difference in the true and that mean age and divided by the true age. And as expected, as fossil age estimates are much closer to the true value, like as the fossil age ranges are much more precise, we get those age estimates much closer to those true values. Though it's also reassuring to see that even when we have very imprecise age ranges associated with the well-dated fossils and 50% of our fossils being poorly dated, we still, for the well-dated fossils, get a very low amount of error in our mean age estimates. When we look at this pattern for the poorly dated fossils, so the fossils that all just fall within this poorly dated deposit, we also see a predictable pattern. So in this case, we see a decrease in the coverage and an increase in the rate as we increase the amount of, or the proportion of fossils that are fall within this poorly dated deposit, as well as when we increase the uncertainty in the more precisely dated fossils. And this is as anticipated, and even though we have low coverage in some of these data sets, we do see that overall the error rate in our mean age estimates is still relatively low. So overall, our simulations indicate that for fossils found within this deposit that has this really diffuse age range, the information contributed by fossils in more precisely dated formations and the morphological characters that are present within those poorly dated fossils can provide us information to get more precise estimates for the formations or for those fossils themselves. And so, like all simulations, ours are actually quite optimistic. And so we wanted to use an empirical data set to validate this approach for dating these poorly dated formations or imprecisely dated fossils. And so we used the data set that I just mentioned before from the study by Thomas et al. To compare the estimated ages for fossils that fall within a hypothetical poorly dated formation. And so to do this, we assigned essentially half of the fossils to this poorly dated deposit and increased the uncertainty in their age range than compared to what we get from the actual fossil record. So what this figure is showing is the ages for just the ages associated for just the fossils that fall within our hypothetical poorly dated deposit. And the prior here is showing how we extended the uncertainty for these fossils to be between about 14 million and 44 million years age range. So in reality, all of these fossils have much, much more precise age ranges that are observed from the dated formations where they come from. And these are shown in pink for each of these. And so in all of our comparisons, what we're seeing is that the marginal posterior 95% credible intervals overlaps with our observed age range for each of these fossils where we extended the uncertainty for their ages. And so what we're showing here is the blue marginal represents the observed or the estimated posterior of each of these fossil ages. And interestingly, there are a few fossils. So while we do see concentration of the marginal posterior, at the observed age range, we do see a subset of fossils where the marginal posterior remains really quite diffuse. And so for some of these, at least for some of the ones that I've looked at, these fossils are represented by very incomplete morphology. So these are fairly incomplete fossil specimens. And so what's cool about this is that what we're really seeing is that when we have precise information coming from one source, say from the fossil record, that can lead toward better information in dating and essentially almost even and potentially even the placement of those fossils. And the same is true in the reverse. If we have more information in the morphological data, and even with the diffuse time information associated with that fossil, we can then better inform our phylogenetic analyses overall and lead toward more robust and reliable inferences. So essentially, these poorly dated formations are collections of poorly dated fossils, like ones coming from the Gobi Desert deposit, Cretaceous deposit in Mongolia, or the Baltic Amber. These contain lots of important information about the ancient lineages that are preserved in those formations. So by harnessing these different sources of information, the morphology coming from various fossil and living taxa, the molecular data, and as well as the age information from these various fossil specimens, we are able to essentially estimate, have better estimates of the timing and the diversification processes of the taxa we're interested in, but potentially also narrowing our understanding of the timing of those fossils themselves and the ecological and historical context in which they lived. And so this is exciting to us because it kind of gives much more a way to more robustly understand both fossils and living taxa in a more holistic way. So all of the studies that I've shown so far have really considered fossils as individual specimens that are preserved in the fossil record. However, this sort of specimen or individual specimen based type data sets for fossils are relatively uncommon. And so instead, fossil data are often curated and collected and described as stratigraphic ranges. And so in this case, there may be some fossil species that are known from only one specimen, where others are known to be found from specimens in different formations or different time intervals. And these are essentially described as stratigraphic ranges. For these fossil species spanning time intervals or multiple time points, they are essentially condensed into a single taxonomic lineage where that's dated essentially just by the appearance or the age associated with the first taxon representing that lineage and the last taxon representing that lineage. So essentially, we came to understand that it's going to be really important to account for the nature of the collection, curation, and use of the fossil record, because it's not quite exactly going to always be in this sort of specimen level type of format. So this figure shows these types of stratigraphic ranges for several genera in a clade of brachiopods, and a few of these genera are known from only a single specimen, whereas others are known to extend across multiple time points. And so paleobiologists have used for a long time data sets just like this to understand various patterns of diversification and preservation in the fossil record. And so this gives us, these types of patterns give paleobiologists a lot of information about not just the origination and extinction of particular lineages, but about the preservation and recovery biases that may impact our understanding of lineage diversity. And so it's really worth recognizing that we have a wealth of data sets that are just like this, housed in paleobiological databases that might not also have information that can be easily analyzed in a complete phylogenetic framework. So our interest was to consider these data sets as well from that kind of phylogenetic perspective, even though we might not have lots of information to estimate phylogeny. And so through our collaboration with Rachel Warnock, who's a theoretical paleobiologist, we've extended these fossilized birth-death processes to account for essentially the way that the paleontological data are collected, summarized, and maintained. And so this is an extension of the fossilized birth-death process that we call the fossilized birth-death range process, and accounts for, it's essentially a process that gives rise to a phylogeny with sampled fossils that also have stratigraphic ranges. And so we generated a mathematical framework for calculating the probability of this tree with these ranges given a set of macroevolutionary parameters. And so this is described in a paper that came out a couple years ago where we described this fossilized birth-death range process. And so in this FBD range process, what we have is a model that describes a generating process for a set of observations that are represented as stratigraphic ranges. So here we have, in this cartoon, we have our first and last appearance of stratigraphic ranges and some singletons. And so what the model does is it accounts for the fact that there are lineages that are unobserved. So just like when we have our full phylogenetic process, we have a complete tree where we don't observe those lineages that don't have sampled descendants. And so the same is true here, where we have lineages that are unsampled, or parts of the lineages that are unsampled. And so just like with our regular fossilized birth-death process, we're accounting for this complete process, for very much of which is not observed. And so for every sampled lineage, the model then marginalizes over the extended stratigraphic range. So we even if we observe part of a lineage, we then can marginalize either analytically or algorithmically the extension of that range that we don't observe. And so in the end, we can also marginalize over the underlying relationships among these lineages. And even this is the case, even if we might have a data set where we don't have the information to actually infer these relationships. And thus, this model can be used to account for the structure in the paleontological data, but also in the case that we do have that information for building phylogenetic trees, then it can become useful for fully integrating these stratigraphic ranges into these phylogenetic approaches too. So I'm just checking the time and seeing that we're getting kind of close. So I'm probably going to skip a couple things, but you know, essentially just want to sort of wrap this up, that we're continuing to work on various approaches of using these types of pure paleontological data sets to apply to these data sets where we have just a portion of the data from the fossil record. And so these approaches are commonly used, like historically in paleontology. Paleontologists will apply these various summary statistic approaches to these types of data sets to understand these processes. And so with this fossilized birth-death range models, we now have ways of accounting for this underlying phylogenetic process, but in addition, these summary statistic methods don't really account for the sampling process when you have singletons and other things in your data set. So the fossilized birth death range model really enables us to integrate our understanding of fossil recovery in a much better way than previous approaches had. And so since we're kind of getting a little bit short on time, I'm going to just sort of mention some of the things that we're working on currently. So we recently published a study looking at the performance of these methods for estimating speciation and extinction events of or speciation and extinction rates in this process. And so now we're extending this to account for the heterogeneous or time heterogeneous process, so we can estimate the change in fossil sampling, so fossil recovery rates, extinction, and speciation over different time intervals in the fossil record. And this is work that is done in collaboration with Walker Pett and Rachel Warnock. And so we hope to have this completed soon. And so essentially what I wanted to kind of give an overview of today really was like this evolution of our understanding of modeling the fossil record through this fossilized birth death process model. And so with this, all of the things that we've kind of come to here has led us to kind of see this birth death process as sort of a model family. And so we have these different ways in which we can apply this model to different kinds of data sets and different kinds of questions. And so we have this full specimen level fossilized birth death process where each observation back in time represents an individual specimen. And we have this fossilized birth death range process that can also be integrated into a phylogenetic framework where we have some singletons and some lineages represented as fossilized or stratigraphic ranges. And these are both useful for when we have data sets of fossil sampling times, as well as the phylogenetic character data. But we also have this approach can be useful for when we have a data set where we don't have the phylogenetic character data and only have the occurrence times coming from the these paleobiological databases or really explicit fossil records of different groups. And so when we have these purely paleontological data sets, we can also use these approaches to understand origination and extinction dynamics, as well as the process of fossil recovery and how that's changed over time. And I'll also mention is that variants of this fossilized birth death model family have also been applied in the analysis of infectious diseases, as well as in the study of linguistics, which are some of these applications are really interesting. And so there's quite a lot of ongoing work that's being done on these types of models and approaches for understanding macroevolution and infectious disease. It's currently being done by a number of people, including people with whom we collaborate and who've been part of my lab. And so I think that there's a lot of promising stuff that will be coming out soon for this. And so all of the methods that I've described today are currently available in the program RevBayes, which I think many of you have heard a lot about yesterday and today probably. And this is just to say that RevBayes is a really rich project that's currently ongoing, lots of development. This is just an animation of our Git repository recently. And so there's really a lot of credit goes to the people who are working to implement new models and work on the infrastructure of this software. And this is just a picture of our recent, or actually a couple years ago hackathon that was taken by Sebastian Hona, who is really the lead developer and project leader of RevBase. And I'll just also mention we're currently always like maintaining the documentation and tutorials for RevBase. We hope to have a workshop, a virtual workshop happening again, this year that I think we'll announce later this month. So if people are interested in it, we'll be generating material for that as well. And so with that, I'd just like to acknowledge the members of my lab and my collaborators and co-authors, as well as the organizers of this conference for inviting me to give this talk, and of course our funding. And with that, I'd say thanks.